Bismillahirrahmanirrahim. Assalamu alaikum. This is me Nazir Ahmad from Pakistan. Today our topic of discussion is volume of distribution, apparent volume of distribution and determination of volume of distribution. If you are new on my YouTube channel, please subscribe to my YouTube channel and press the bell icon button so that you may not miss any upcoming video. What is volume of distribution? The volume of distribution, also known as apparent volume of distribution or VD, can be thought of as the fluid volume that is required to contain the entire drug in the body at the same concentration measured in the plasma. It is calculated by dividing the dose that ultimately gets into the systemic circulation by the plasma concentration at time 0 C0. Vd is equal to amount of drug in the body divided by concentration at time 0 C0. Although volume of distribution (Vd) has no physiologic or physical basis, it can be useful to compare the distribution of a drug with the volumes of the water compartments in the body. Distribution of drug into the water compartments in the body. Once a drug enters the body from whatever route of administration, it has the potential to distribute into any one of three functionally distinct compartments of body water or to become sequestered in a cellular site. Plasma compartment. If a drug has a very large molecular weight or binds extensively to plasma proteins, it is too large to move out through the endothelial slit junctions of the capillaries and thus is effectively trapped within the plasma vascular compartment. As a consequence, the drug contributes in a volume, the plasma, that is about 6% of the body weight, or in a 70 kg individual, about 4 liters of body fluid. Heparin shows this type of distribution. Extracellular fluid. If a drug has a low molecular weight but is hydrophilic, it can move through the endothelial slit junctions of the capillaries into the interstitial fluid. However, hydrophilic drugs cannot move across the lipid membranes of cells to enter the water phase inside the cell. Therefore, these drugs distribute into a volume that is the sum of the plasma water and the interstitial fluid, which together constitute the extracellular fluid. This is about 20% of the body weight, or about 14 liter in a 70 kg individual. Aminoglycoside antibiotic shows this type of distribution. Total body water. If a drug has a low molecular weight and is hydrophobic, not only can it move into the interstitium through the slit junctions, but it can also move through the cell membranes into the intracellular fluid. The drug therefore distributes into a volume of about 60% of body weight, or about 42 liter in a 70 kg individual. Ethanol exhibits this apparent volume of distribution. Apparent volume of distribution. A drug rarely associates exclusively with only one of the water compartments of the body. Instead, the vast majority of drugs distribute into several compartments, often avidly binding cellular compartments such as lipids abundant in adipocytes and cell membranes, proteins abundant in plasma and within cells, and nucleic acids abundant in the nuclei of cells. Therefore, the volume into which drugs distribute is called the apparent volume of distribution, or VD. VD is a useful pharmacokinetic parameter for calculating a drug's loading dose. Determination of VD. The fact that drug clearance is usually a first order process allows calculation of VD. First order means that a constant fraction of the drug is eliminated per unit of time. This process can be most easily analyzed by plotting the log of the plasma drug concentration (CP) versus time. The concentration of drug in the plasma can be extrapolated back to time zero, the time of injection, on the y-axis to determine C0, which is the concentration of drug that would have been achieved if the distribution phase had occurred instantly. This allows calculation of VD as VD is equal to dose divided by C0. For example, if 10 mg of drug are injected into a patient and the plasma concentration is extrapolated back to time 0, the concentration is C0 is equal to 1 mg per liter, and then VD is equal to 10 mg per 1 mg per liter is equal to 10 liter. Effect of VD on drug half-life. A large VD has an important influence on the half-life of a drug because drug elimination depends on the amount of drug delivered to the liver or kidney or other organs where metabolism occurs per unit of time. Delivery of drug to the organs of elimination depends not only on blood flow but also on the fraction of the drug in the plasma. If the VD for a drug is large, most of the drug is in the extraplasmic space and is unavailable to the excretory organs. Therefore, any factor that increases VD can lead to an increase in the half-life and extend the duration of action of the drug. Note: an exceptionally large VD indicates considerable sequestration of the drug in some tissues or compartments. In the next presentation, I will present drug clearance through metabolism, kinetics of metabolism, reactions of drug metabolism. Thank you for watching this presentation. If you have any question, please write in the comment section below.